Hello everyone, this is the last lecture on our presentation on gluconeogenesis, glycogen metabolism and the pentose phosphate pathway, and in this lecture we're going to look at the pentose phosphate pathway. We will look at the purpose of this pathway, its regulation, and of course the specific reactions that take place within this pathway. So one of the main questions we want to answer in this section is, can glucose also provide the electrons that are required for biosynthesis? So NADPH, we know, is the coenzyme that is used for reductive reactions in anabolism, in other words in biosynthesis, and this NADPH is produced here in the pentose phosphate pathway. There are other pathways as well where it is produced, but one of the reasons we look at the pentose phosphate pathway is that it is an important source of NADPH. So this pathway is also important for the synthesis of ribose, which of course is essential in the synthesis of nucleic acids. Ribose and ribose derivatives will eventually be built up into the nucleotides that are used to build nucleic acids. So the pathway consists of two oxidative processes, which is where the NADPH is formed, followed also by 5 non-oxidative steps. All cells actually require the synthesis of ribose, which is essential for nucleic acids, but the oxidative phase of the pentose phosphate pathway operates more actively in certain cells than in others. In other words, it's more active in, for example, the liver and adipose tissues. So it is tissue distribution that quite often determines how the function of the pentose phosphate pathway. In other words, why the NADPH has to be produced and at what level it has to be produced within a specific cell. So the production of NADPH for fatty acid synthesis in the liver and adipose tissues, it is more important in those tissues than in other cells, so those cells would require more NADPH for those reasons. Cholesterol is synthesized only in the liver, so that also takes quite a lot of reducing power, and it is not required in other cells in mammalian bodies. And for example, steroid hormone synthesis in adrenal glands, ovaries and testes also require NADPH, and this is of course not a requirement to produce NADPH for these purposes in most of the other cells within mammals. So ATP of course is the energy provider for cells. So ATP will provide the energy for most of the reactions where energy is required for biosynthesis. But cells also need reducing power. In other words, it also needs electrons that can be donated onto a specific product during biosynthesis. NADPH and NADH are not interchangeable for these purposes. We've discussed this before, and NADH of course participates in using the free energy of metabolite oxidation in order to synthesize ATP. That's what we've discussed quite at length. The NADH from glycolysis and from the TCA cycle. Those electrons are carried onto the electron transport pathway, takes part in oxidative phosphorylation, which eventually helps the ATP synthase to function and produce ATP. But the NADPH uses the free energy of metabolite oxidation for biosynthesis, so it'll grasp these electrons, these high energy electrons from metabolites that are being degraded, and it'll carry those over into reactions of biosynthesis where reducing power is needed. So the pentose phosphate pathway generates NADPH from the oxidation of glucose 6-phosphate. It is, in other words, an alternate to glycolysis. So the first reaction actually takes place, of course, is glucose comes into the cell, it gets phosphorylated by hexokinase, and it produces glucose 6-phosphate, and from here it can either carry on into the pathway of glycolysis that we've discussed at length, or it can be shunted towards the pentose phosphate pathway depending on the regulation of the cell. So I put this image up here. The structural information is of course not important for exam purposes, but I wanted you to appreciate the subtle difference between NAD and NADP. It's really just a phosphate group, but that does make all of the difference. This image also shows where the electrons are carried in the form of a hydride. So even though this is a very small and subtle difference between the structures of these two coenzymes, they are not interchangeable, and NADPH participates in catabolism, whereas NADPH participates in anabolism. So let's look at the pathway. It has an oxidative phase where the reactions that will produce NADPH takes place. This is an irreversible and linear phase. And this is essentially what it looks like. So we have, of course, glucose is turned into glucose 6-phosphate by hexokinase, and as we said, then it can be shunted into the pentose phosphate pathway. So glucose 6-phosphate can then be turned into 6-phosphogluconolactone, and that is the first reaction that produces an NADPH. Then we have a hydrolysis reaction by lactonase, which produces 6-phosphogluconate, and then we have a second reaction, which is an oxidative decarboxylation, where the second NADPH is produced, carbon dioxide is released, and we actually have a ribulose 5-phosphate, and it is this that can be fed into the non-oxidative phase of the pathway. The reaction which produces ribose 5-phosphate, this pathway, part of the pathway has reversible reactions which can feed into glycolysis, and it is a cyclical pathway. So a lot of the products of certain reactions are the substrates for the next reaction, but they will then quite often produce products which are metabolites which can take place in earlier reactions in this pathway. For example, purposes for the BTN223 course, you have to know the oxidative phase, enzymes, intermediates, and reactions at the same level of detail that you had to know for glycolysis and for the TCA cycle. We are going to discuss all of the reactions of the pathway, but for exam purposes, it's the oxidative phase reactions and intermediates that you need to concentrate on. So, to the reactions then. The first reaction, catalyzed by glucose 6-phosphate dehydrogenase, catalyzes the net transfer of a hydride ion onto NADP to form, from the C1 carbon of glucose 6-phosphate, and that'll produce 6-phosphogluconolactone. So this is the reaction, of course, where we have our first NADPH produced, and it is catalyzed by glucose 6-phosphate dehydrogenase. It is an irreversible step, and it is highly regulated. The second reaction is the lactonase reaction, where 6-phosphogluconolactonase catalyzes the hydrolysis of 6-phosphogluconolactone to 6-phosphogluconate, and you'll see it as a hydrolysis reaction. And then the third step, the last step of the oxidative part of the pathway, the 6-phosphogluconate dehydrogenase enzyme catalyzes the oxidative decarboxylation of 6-phosphogluconate to yield ribulose 5-phosphate, carbon dioxide, and an NADPH. It's a two-step process that occurs via beta-keto acid intermediate, and the resulting product, the D-ribulose 5-phosphate, is the substrate for the non-oxidative reactions of the pentose phosphate pathway. So here we have our second NADPH produced, and we have the ribulose 5-phosphate, which will now be fed into the non-oxidative reactions of the pentose phosphate pathway. So, a very brief summary. There's three reactions in this oxidative phase. Glucose 6-phosphate is turned into 6-phosphogluconolactone, and an NADPH is produced. And in the third reaction, we also have an NADPH produced when 6-phosphogluconate is turned into ribulose 5-phosphate and a carbon dioxide. So this generates two molecules of NADPH from a glucose 6-phosphate. In the non-oxidative steps, we have the first reaction catalyzed by Phosphopentose isomerase, which converts ribulose 5-phosphate into ribose 5-phosphate. And this ribose 5-phosphate that is produced is the most essential precursor for nucleotide biosynthesis. So the ribose 5-phosphate can now be used to create these different NTPs that are required for NTPs and DNTPs that are required for making either RNA or DNA. The reaction that takes place here is that a ketose sugar is turned into an aldose sugar. The fifth reaction is catalyzed by Phosphopentose epimerase. It converts ribose 5-phosphate into xylulose 5-phosphate. It doesn't appear if too much is happening here, but of course it changes the enzyme changes the stereochemistry of the ribose 5-phosphate in order to produce a xylulose 5-phosphate, and changing the stereochemistry, beg your pardon, is what an epimerase enzyme does. The sixth reaction is a Transketolase reaction, which catalyzes the transfer of a 2-carbon unit from xylulose 5-phosphate to ribose 5-phosphate, resulting in glyceraldehyde 3-phosphate and acetoheptulase 7-phosphate. So we have a 2-carbon carried off from the xylulose 5-phosphate. It is added onto a ribose 5-phosphate, which of course was an earlier product in the pathway by the Transketolase, and this produces a glyceraldehyde 3-phosphate and a acetoheptulase 7-phosphate. Now, this glyceraldehyde 3-phosphate is going to be important in the next reaction as well, even though of course you will recognize this as being an intermediate of glycolysis. In the seventh reaction, the second last reaction, a Transaldolase transfers a 3-carbon unit from that acetoheptulase 7-phosphate produced in the previous reaction onto the glyceraldehyde 3-phosphate also produced in the previous reaction, and this will yield erythrose 4-phosphate and fructose 6-phosphate. And it is, as I said, a Transaldolase reaction. So the final reaction will then take the erythrose 4-phosphate and it'll transfer a 2-carbon unit from xylulose 5-phosphate, which is of course a product of the Transaldolase reaction earlier in the pathway. It'll take that 2-carbon unit and add it to the erythrose 4-phosphate in order to produce a fructose 6-phosphate and a glyceraldehyde 3-phosphate. And what we see here now is that these two products that are produced at the end of this pathway can either be fed back into the pathway, or of course they can be fed into the glycolysis pathway, because they're intermediates of glycolysis as well. And this brings us to the point of how the pentose phosphate pathway is regulated. So how glucose 6-phosphate is used in the cell depends on the cell's need for ATP, NADPH, and ribose 5-phosphate. So glucose can be a substrate of either glycolysis and pentose phosphate pathway. As I said, they can be shunted into either of the two pathways, the glucose molecules, and the choice of which pathway is used will depend on the relative need of the cell for the biosynthesis and for energy metabolism. ATP can be produced, of course, if glucose 6-phosphate is sent into glycolysis. We can have glycolysis, the TCA cycle, oxidative phosphorylation, and ATP will be produced. But NADPH and ribose 5-phosphate can also be produced if the pentose phosphate pathway is used instead of glycolysis. So depending on the needs of the cell, the reactions of glycolysis and pentose phosphate pathway can be combined in any one of 4 ways. So I just wanted to show this slide that shows the entire pathway again, because that'll help us make a lot more sense of the next slide. So the 4 ways in which the regulation can take place for us to be able to utilize either the glycolysis or pentose phosphate pathway is if the cell needs both ribose 5-phosphate and NADPH. The first 4 reactions of the pentose phosphate pathway will be dominant, and NADPH will be produced, and ribose 5-phosphate will be the principal product of carbon metabolism. So what that means is that the first 4 reactions, the 3 oxidative pathway reactions and the first reaction of the non-oxidative pathway will predominate. Then in the second condition, if more ribose 5-phosphate than NADPH is needed, then synthesis of ribose 5-phosphate will be accomplished without making a lot of NADPH by bypassing the oxidative reactions of the pentose phosphate pathway. Now, how can we simply bypass the oxidative phase? Remember, we don't have to start this pathway with glucose 6-phosphate, because of the cyclic nature of the non-oxidative phase, we can actually start the reactions from fructose 6-phosphate and glyceraldehyde 3-phosphate and allow these reactions to run in the synthesis direction, in other words, to produce our ribose 5-phosphate in this way. Then if more NADPH than ribose 5-phosphate is needed, it can be accomplished if ribose 5-phosphate is produced in the pentose phosphate pathway and then redirected to produce glycolytic intermediates. So some ribose phosphate is needed, but the excess ribose 5-phosphate will then be redirected towards the glycolytic intermediates, glyceraldehyde 3-phosphate and fructose 6-phosphate, and that'll allow the production of more energy. And then finally, if both NADPH and ATP are needed but not ribose 5-phosphate, then this is done by redirecting all of the ribose phosphate that is produced into producing fructose 6-phosphate and glyceraldehyde 3-phosphate, and that is fed into glycolysis to produce ATP and pyruvate, and of course the pyruvate will go on to the TCA cycle and electron transport. So that brings us to the end of this lecture, the end of the presentation on gluconeogenesis, glycogen metabolism, and pentose phosphate pathway.